Hello, my name is Glynn and I'm in Group 2B for Tissue Engineering. The title of our poster is Determination of Mechanical Properties, Degradation Rates and Cellular Viability of Gelatin-Based TCP Scaffolds. Gelatin is a natural biopolymer derived from collagen. It's commonly utilised with beta-tricalcium phosphate to produce nanocomposite scaffolds. This study aims to assess differential characteristics between pristine gelatin and gelatin-based TCP scaffolds to determine the suitability for bone tissue engineering strategies by assessing mechanical properties, degradation behaviour and cellular viability. Our hypothesis states the addition of beta-tricalcium phosphate-based TCB is gelatin scaffold seeded with MC3TCE1 osteoblast cells creates a biopolymer scaffold with unchanged mechanical and biological properties. In order to carry out the experiment, we had to prepare our gelatin-based TCP scaffolds using freeze-drying fabrication. We then carried out tensile testing on the rehydrated scaffolds to determine the compressive strength. We assessed the swellability by hydrating the scaffolds with ethanol and PBS, then incubated them for 7 days. Following this, we reweighed them and determine the degradation rate. The scaffolds were then seeded with MC3T3E1 pre-osteoblast cells, and their viability was tested using host DNA assay and were quantified by measuring fluorescence intensity. In terms of results, addition of TCP to gelatin scaffolds increases resistance of the scaffold to elastic deformation under a load. This is illustrated by higher Young's modulus value as seen in Figure 4 and Figure 5. TCP has a marginally higher degradation rate, although both demonstrate high rates. As seen in Figure 6, both gelatin and TCP demonstrate high degradation rates, with TCP displaying 100% degradation and gelatin displaying 96% after 7 days. From the standard reference curve, the unknown DNA concentrations from the seeded cells were determined at 485 nanometers. The concentrations were then converted to cell number, which is representative of cell viability for TCP and gelatin scaffolds, at 1:10 and 1:20 dilutions respectively. The gelatin TCP has a greater cellular viability than the gelatin. Upon statistical analysis, the results were we obtained a p-value of less than 0.05, which allows us to reject our null hypothesis. This may imply that beta-TCB alters mechanical and biological properties of scaffold. However, a larger sample size would be required for a more conclusive result.